Thank you, Gabriella, for the nice introduction and invitation to the meeting, and thank you, Sharon. Um, when your name got mentioned like 5, 10 times, you get this strange image of 10-foot tall Greek goddess or god. But I'm sorry I disappoint you. I have to apologize for that. The other thing I want to apologize, I have a recovering from cold, so I may be coughing along the way, but hopefully that's not going to affect your attention. Maybe that will draw your attention too, because we're very close to lunch hour, right? Okay, well, people talk about this. The advantages of the last person is that now you know all about how wonderful the fermented foods are. But here are a bunch of fermented food. Some of them have been around for a long, long time. For example, honey and yogurt has been known as food of God. And we all know that if you have a nice bottle of Bordeaux and a pint of Guinness, that would make you very healthy. But the problem is we don't know what's in there is good for you. We don't know how it's good for you. We don't even know when it's good for you or where it's good for you. So that's the big problem. But the other things that kind of help us is some of the new technology. You could start hearing these 16S sequencing and things like that. It all happened in the last 20 years or so. For example, you have PCR, polymerase chain reaction. It increased the sensitivity, you increase the throughput, and then save a lot of time because if you're as ancient as me, we used to do northern blot, and nobody know what's northern blot anymore. Then you have the microarray. It's just about 10 years ago, but it's quickly moved through high throughput sequencing, or now people call it the next generation sequencing. I don't quite understand what's that next generation's for, but it's called next generation sequencing. That's where you get RNA sequencing, or you hear things like metagenomic sequencing, and then 16S sequencing and things like that. You can also do splicing, methylation, SNP analysis, and really expand on the genomic study. At the protein level, you also have multiplexing assay, allow you to do detection of multiple number of proteins. It could be 27 cytokines at the same time. So you have a lot of tool. And the more important thing is that you have the software, the computer technology to put everything together. In the old days, we look at one gene at a time, and now you look at thousands of genes and want to look at how pathways interact with each other. So this becomes very important. So you have all these new tools to look at things at the mechanistic level. But before we go into the mechanistic, I think the first thing you need to look at is what actually in the food itself. Try not to read anything. I'm just showing this as an example. For example, in the type fermented rice product, you can have 10 or 20 different bacteria in there. So it's relatively complicated. And if you look at, what is this one? Kefir grains. It's also very complicated. I want you to focus on the bottom one. The bottom one, they're from different source. So you can see that depends on the source, the bacteria family are also very different. So that's one complexity you have to think about. This is from fermented soybean paste. And this is from 16S ribosomal RNA sequencing. And focus on the bottom one over here. Again, these are from different sources. If you look at distribution of bacteria, which is color-coded, you can tell that from different sources, they are very different from it. So you need to define your source first. In addition to bacteria, you also have the so-called plant product. This is just an example from the soy germ. You have the isoflavones. You also have saponin, phytosterol, and tocopherols. These are all many different compounds. They're different structure. And another thing you want to look at is that, for example, this one over here, the isoflavone with the glycoside attached to it, after fermentation, the white bar, you have the aglycol form pop up. So that's another thing that you need to think about. All these different phytochemicals, could be thousands of them, what actually in your fermented product. That's important to know. So now we go into the mechanistic level. And of course, the first thing, you have bacteria in there. Obviously, you need to look at microbiome, right? So this is just give you some number of what the complexity of human microbiome, or more specifically, the gut microbiome. So the bacteria outnumber human cells by tenfold. So you can see over here it's like 10 to the 14 versus 10 to the 13. For the bacteria, you have like 3 million genes in there you need to look at versus like 30,000 genes for a human. You have about 500 to 1,000 species of bacteria, and you can divide it into 99% of it is 30 to 40 different species. And you can also look at colon. The bacteria make up most of the flora in colon, and it's about 60% of, if you fecal sample come out, it's 60% is bacteria. So that's a lot of poop, right? What's more important is the interaction of microbiome with the host. It can modulate inflammatory responses. It can synthesize small molecule and then change protein or more complex molecule to a smaller one and get taken up by the host. And also can change the energy availability in the diet. But the first thing you need to look at is, you have microbiome, you have bacteria in there, but could you change anything from based on diet? And the answer seems to be yes. This is just comparison of African village kids versus the EU kids. So you can see that the green bacteria is much more in the African kid, and the EU kid tend to have the red bacteria or pink bacteria much more. It turned out that these bacteria are firmicutes, and these are bacteroids. So it depends on what you eat. You're going to have a very different profile for microbiome. And yes, if you eat something, it'll change. I want you to focus on this over here. This is just eating from 0, 1, and 8. And you can see the biggest changes after 8 weeks. The bacteria patterns percentage are very different. So yes, you can change it if you have a fermented food such as kimchi or yogurt or anything. It will change that. Now, what are those changes going to do? And this is actually a relatively difficult talk for me to do because it is everything under the sun. And it's kind of hard to tell you everything under the sun. So I'm just going to be very close. What's that?
On purpose, we asked you to do this because we knew that it's everything under the sun, as you said.
This is not everything under the sun, but if you want, you can go to the review for everything under the sun. But the first thing, most prominent things that people talk about for these gut microbiome is the modulation of host immune system. But itself is relatively difficult, complicated. It can do multiple things. For example, if you look over here, it can have a local effect in the intestinal mucosal area. So it's caused inflammatory responses in mucosa. So if you have changes in perturbation of the microbiome, it causes the inflammation. Now, things may get a little bit more complicated because of this over here. if you have changes in gut microbiome, you actually have an effect at the remote side. In this case, there's something called microbiome asthma theory. So if you have a perturbed microbiome, actually it develops more asthma. So this is already, you have mucosal changes, you also have indirect changes. Here you have a high-fat diet scenario. High-fat diet is known to changes the microbiome. And we know that it will change multiple tissue at multiple tissue level. For example, increased liver lipogenesis, but also induce inflammation of multiple tissue, because the theory is that these high-fat diet can increase the gram-negative bacteria, increase production of the lipopolysaccharide, which is an inflammatory agent. So you have a sub-level of inflammation. So it depends on what you eat. You may have sub-level inflammation, you may not. You may have localized intestinal inflammation. You could also have a remote side inflammation. So things are relatively complicated just by changing microbiome. And then you have all the other compound in the diet that, once you go past the gut, it'll go into the it will go into the circulation. And I will try to lump everything together. I think most of it will be nutrient-gene interaction. Why? Because we now know that gene is an on-off switch for a particular pathway. It also gives you the so-called efficacy, the biological effect. And we also know that nutrient induce gene involved in a very critical pathway, including hormone, cytokine, cell number homeostasis, or xenobiotic metabolism. And the other thing is that gene can confer or modulate nutrient efficacy. I'll talk a little bit about it a little bit later. And here you are again. Instead of microbiome, you are what you eat. I'm looking in terms of diet, because your diet can change all these things and eventually causes some kind of biological effect. So the first thing is genetic regulation, basically a regulation of the transcriptome. I'm just going to use my own data as an example. This is not specific for a particular fermented product, but we do look at genistein, which is from soya. You know that fermented soya should have a glycone form of genistein. These are some of the typical pathway that we look at. For example, this marker over here, CDKN1A, is for cell cycle inhibition and DNA damage. We also have CYP1A1 and NQO1. These are xenobiotic metabolizing enzyme. And in this case, we're looking at the prostate cancer cell line. So we have prostate-specific antigen, which is indicated for the androgen-dependent pathway. And you also have the AR, which is androgen receptor. You can see over here, the message is that the more red, you have the increase. The green is downregulation. So you can see that here are a bunch of different compounds. They all act very differently. In some cases, you have activation of, for example, CYP1A1 over here. In some cases, there's nothing going on. So individual compound, or what you get into the system, become very important. The other thing would be concentration. You can tell some of the compound can occur at a very low concentration, but some you need to have relatively high concentration to cause an effect. So this is some example for a diet-gene interaction. So you can think that if you have a food-derived compound, for example, activate cytochrome P450-1A1, which is a xenobiotic metabolizing enzyme, it can help removal of carcinogen, which is good for you. But on the other hand, if you have individual with different phenotype for cytochrome P450-1A1, this ability will be changed. Therefore, your ability to elicit a response will be different. Okay, so the message here is that a compound can act on multiple pathways once you get into the cell. And the other one is it's important to identify a particular target tissue and system because you have so many different pathways and tissue you need to look at. Again, this is another one of our own study. We look at ECOR, which is a microbial degradation product from daisy, and genosine. This is an older study. This is using a microarray analysis. Again, green is downregulation, and red is upregulation. Just focus on the top panel A over here. All these genes, which you cannot read, they're from the androgen-dependent pathway. So what these compounds are doing to these genes is to downregulate the androgen-dependent pathway. Why is this important? Because exposure to androgen is known to be risk factor for prostate cancer. So you would think that maybe eat soya would be good for you in terms of protect against prostate cancer. And this may be a potential mechanism that would work. This is a study with genosine, and again, it's a microarray study, but we kind of compile everything and look at what are the ask the question, what are the concentration that you can see for activate multiple pathway? You can see over here that androgen response gene occurred at much lower concentration. IGF-1, which is known to be a proliferative agent in cancer, means bad. So inhibition is occur at a much lower concentration. But for DNA damage and stress response occur at much higher concentration. And this is the dietary concentration. I'll come back to that dietary concentration. I'm just planting a seed here. So you already have all these genes. You have like 30,000 genes to look at. There are more things to look at. Something called epigenetic or epigenomic, because everybody wants to have omic now, full omic, whatever omic. So this is stably, I'm just getting from Wikipedia the definition, stably inheritable phenotype resulting from changes in chromosome without alteration in the DNA sequence. What does that all mean? Well, it can be something like this. For example, you can change the methyl group, methylation pattern of DNA, the chromosome. That can cause activation or suppression of the DNA. Another thing you can do to the DNA is to histone modification. It could be methylation or acetylation, and it could be repressing or activating a certain gene. So all these does not involve changing the DNA sequence. Okay. And this is just some of the compound from different veggie and things like that, and it's been documented that it can change the epigenetically. So that's again. That's a potential mechanism that you can look at in terms of fermented food. But things become even more complicated. There's something called a small RNA now. It's here it says mRNA, but your body and even plants produce these small RNA. What it does is actually can regulate gene expression. So in the old days, you have the one gene, one product kind of thinking process. But now the gene regulation is much more complicated. In this case, for let's say that the animal, you can cause a suppression of the gene, suppression of gene. But consider that these small RNA are also in the plant. In your fermented food, there will be small RNA. What does it do? Does it do anything to the cell or people or what? there's maybe the last couple of years, there's one Chinese group that demonstrated that you can actually isolate rice, small RNA, and have a physiological effect. So that add another layer of complexity to what we're talking about, because we have the microbiome, we have whatever compound that's in there, thousands of them, but now you have the microRNA kind of doing something to it. This happened to be one of Sharon Ross' paper, review paper, but it's very nice. I'm just kissing up to you. Anyway, there's several compounds that actually causes changes in small RNA. But even more complicated now. We cannot just stop at the small RNA. There's something called in utero and transgenic effect. So be careful. What you eat now may pass along to your grandkids. Okay, don't think very lightly when you go to lunch today and want to look at that big piece of meat. Want to eat that? Think twice. Okay. There are some study looking at genistein, which can changes the color code over here. This is over here that seem to be related to methylation pattern for DNA. And other people have shown that if you have a high-fat diet, you could actually pass on to multiple generations. So something to think about very carefully. I'm going to try to finish off. This has nothing to do with mechanism, but I think it's important to think of it, the human, as a relatively complex organism. And you have individual difference. This is our own study in collaboration with one of my colleagues, Bev Cliveden. We look at responses of beta-carotene within different individuals. So these are free-living individuals. We give them watermelon juice or tomato juice and look at what happened after 4 weeks later. Collect blood and look at beta-carotene circulating level. You can see over here, some people never go up. The circulating level never go up. But some people go up like 5, 6-fold. If you even have similar concentration, some stay the same, some go up like crazy. Individual difference. Fermented food for everybody, we don't know. I mean, it may depend on individual difference. And then you have the timing and physiological concentration issue. This is a study looking at the folate. Want you focus on the 3 weeks. You get this inhibition. If you introduce folate at 3 weeks, you get this inhibition. But if you wait till 6 weeks and introduce the folate, look at what happened. You have much higher level of tumor form. So when you get it, you can say that, oh, yogurt is so good for you, but does it mean that you have to get it before you're born, or right after you're born, or before you turn 50? Well, that's a question we need to address. And then finally, is the physiological concentration. You always seen this slide. So this is where dietary concentration, or at least circulating concentration, the highest people report that you can get. So I would argue likely that these pathways may not be relevant in terms of looking at mechanistically, because you won't get that high. You can only stay here. This is, again, our own study with resveratrol on cell level. You have the CDKN1A, which is an indicator for DNA damage. You only can get it up to relatively high concentration and 25 micromolar to see an effect. But if you look at this dotted line over here, which is PSA as an indicator for androgen-dependent pathway, you can get it at relatively low concentration of like 1 or so. And this is androgen-dependent pathway. We actually go off and measure the tumor level and plasma level. You won't get higher than 5, at least in our study. So concerning the mechanism, this may be, the PSA androgen-dependent pathway may be the more relevant pathway. So I'd like to summarize everything. So diet-derived compound can regulate microbiome, can regulate gene expression at genomic level, at epigenomic level, and it can affect cellular pathway. There are many, many cellular pathways you can think of, including hormones, cytokines, biotic metabolism. I think that's a mess. I must be thinking about the grilled meat when I was typing this. It's called meat metabolism. I do eat veggie a lot because I work for USDA. And of course, there are multiple mechanisms can be involved. One important thing to think about, individual may respond to nutrient differently. So a genotype may influence not only the bioavailability of the individual compound, efficacy, and also susceptibility. And then timing and physiological concentration are also important to think about. You may be introduced at the wrong time, cause some big problem later. I also like to show this. What is this? This is the E. coli metabolic pathway, okay? Yeah! So if you think about, you and I, we're multicellular organism, multi-tissue organism. Individual is very different. Wow, this is going to be a little bit more complicated. So I'm just going to say that something about, nutrition research. This, I kind of take steal it from Leroy Hood because he talked about P4 nutrition, medicine. So I changed all the medicine into nutrition. Okay. So I think the nutrition for 21st century, at least the Holy Grail, it will be predictive nutrition, preventive nutrition, and personalized nutrition, and also will be participatory nutrition. Since nutrition, most times you have dietitian and things like that, and also your parents would educate you on what to eat, like my mother. Participatory nutrition is there, so there will be a lot of discussion. We don't have to worry about this. But if you look at it, these are all the things that the Holy Grail that we're looking for, because we know, we decipher the human genome. We should have a roadmap, and we're going to go where no one has gone before. And this is just a knowledge, a bunch of people that contribute to whatever work that we've done. And without them, I couldn't be here. Thank you very much for your attention. And eat your veggie.
Do we have time for one or two questions for Dr. Wang? Anyone in the back? Dr. Bo? Or Ho?
Bo will be fine, thank you. Hi, Thomas. Thank you very much for the interesting talk. Just first point is very simple. You said a high lipid diet will generate more of the LPS, which means that the likelihood of increasing the microbiome leading to the gracilicus. Is that true?
Well, it's an animal study that people introduced high-fat diet, and in the circulation they found there's an increase in the LPS level, and so increased LPS is synonymous to increased inflammation.
Okay.
So that is the correlation.
Thank you. Another one, just asking for your comments. Most of the study, in terms of the regulation of gene, the microbiome, you're dealing with one single adenosine or any others. But what do you think? Get your comment pertaining. If we have a combinatorial, meaning more than one, do you think that would be quite different because the response of the microbiome in terms of ratio could also affect the whole thing. So your comment, please.
Yeah. I think that's the question that we constantly struggle with because I'm a cell biologist and I'm a reductionist. We only look at one pathway, one gene at a time. But unfortunately, things are, as I tried to point out, are much more complicated. So in one sense, you may want to base on epidemiological study and identify a flu, then go from that, and then see what happened. The flu may be a preferred way of doing it. But unfortunately, at the end, you still have to look at mechanistically what are the active component, whether there is any interaction. So there's still a lot of things to work on. And if you see some of my earlier slides, some of the fermented food compound, they have very different property. So let's say that you have a Korean soy paste, and then you have the Chinese soy paste. The Korean would say, well, my soy paste is better than yours. The Chinese would say, well, no way, mine is better than yours. But that's the problem because they may be completely different. They may be all good, and they may be all bad. So that's a problem. But then again, I think the whole food approach may be better in this day and age.